Hello students, I am Mrs. Ghodake-Patil working at Swami Ramananda Bharati Vidya Mandir and Junior College, Tasgaon. Today we continue the new point from the topic enhancement of food production, that is microbes in sewage treatment. Actually, a matter carried off in drainage is called sewage. As we know that sewage is a matter that is carried off in drainage. Thus, it includes human excreta, human wastes, house wastes, pathogenic microbes, and organic matter. Then actually the composition of sewage is 99.5 to 99.9% water and 0.1 to 0.5% inorganic and organic matter. That means in the sewage, amount of water is a very large amount. Then see in the diagram, that is the diagrammatic representation of various stages that we are going to study in detail. Primary stage, preliminary stage, then secondary treatment and tertiary treatment, that's all. So sewage treatment process includes 4 steps that are known as the preliminary treatment, primary treatment, secondary treatment and tertiary treatment. All are seen in the diagrammatic representation. Actually, wastewater is first in the preliminary treatment. First stage is screening and second one is the grit settling, that is remove the heavy solids. Then the second step, that is the primary treatment, in which the removal of suspended solid particles and the settled sewage is undergoes. Then secondary treatment takes place in the sedimentation tank or aeration, then sent to the aeration tank. Then it takes place the third step, that is the tertiary treatment, treated with the effluents. Actually, aerobic bacteria in the aeration tank activated sludge and anaerobic bioreactor, that is the digester, anaerobic bacteria and sludge cake to dispose and water is recycled. तर अशा पद्धतीनं ह्या सिवेज ट्रीटमेंट प्रोसेसमध्ये 4 स्टेप्स इन्क्लूड केलेले आहेत. ह्या आपल्याला स्टडी करायचे आहेत. देन नेक्स्ट वन इज द मायक्रोब्स इन एनर्जी जनरेशन. That energy generation is studied with the biogas production. That biogas production is a mixture of methane 50 to 60%, then CO2 carbon dioxide 30 to 40% and H2S, that is 3%, some about 3%, and other gases are also present. Then this biogas is commonly produced from cattle dung, and we study the production of biogas, that is biogas plant, how it works or how actually it is processed. The biogas plant is or production of biogas is included 3 steps. First one is the hydrolysis, or it is also known as solubilization. Second one is the acidogenesis and third one is the methanogenesis. All these 3 steps are important. That means in first step, that is the hydrolysis or solubilization, it is the initial stage. Raw material is mixed with water in equal proportion to make slurry, which is then fed into the digester. See in the diagram, that is the digester. The slurry is fed into the digester plant. Then second step in the solubilization, that is anaerobic hydrolytic bacteria. That anaerobic hydrolytic bacteria such as, for example, Clostridium and Pseudomonas, that hydrolyze the carbohydrates into simple sugars, protein, and these are the products formed by the activity of the bacteria, simple sugars, proteins into amino acids and lipids. That means carbohydrates conversion, simple sugars, proteins conversion, amino acids आणि लिपिड्सचं कन्वर्जन फॅटी ॲसिड्स अशा पद्धतीनं केलं जातं. दॅट इज हायड्रोलायसिस. देन सेकंड स्टेप दॅट इज नोन ॲज द ॲसिडोजेनेसिस. That acidogenesis is the second stage. It is the facultative anaerobic acidogenic bacteria in that stage. Facultative anaerobic bacteria that are acidogenic and obligate anaerobic organisms convert simple inorganic material into acids like formic acid and acetic acid. That is known as the acidogenesis. Besides this formic acid and acetic acid, hydrogen and CO2 also formed. And the third stage, that is known as the methanogenesis. In the methanogenesis, it is the last stage in which anaerobic methanogenic bacteria like Methanobacterium and Methanococcus convert acetate, hydrogen and CO2 into methane. Then we study the benefits of biogas. Actually, biogas is very much beneficial. As we know, it is used for the domestic purposes. Besides these, biogas has number of advantages such as it is a cheap, safe and a renewable source of energy. It can be easily generated and stored and transported. It can be used for domestic lighting, cooking, street lighting as well as small scale industries. It burns with blue flame, that is without smoke. It helps to improve sanitation of the surrounding and it is eco-friendly and does not cause pollution or imbalance of the environment. And the sludge, see here, the sludge which is left over is used as a fertilizer. In this way, biogas is important. Then, next, that is the role of microbes as a biofertilizer. Biofertilizers are the biological fertilizers which increases soil fertility. It is prepared from farmyard manure, that is FYM, farmyard manure, compost and green manure. Biofertilizers are nitrogen fixing microorganisms like bacteria and in that specially cyanobacteria and fungi. Types of biofertilizers. There are number of different types of biofertilizers such as nitrogen fixing biofertilizers. First example, nitrogen fixing biofertilizers. Nitrogen fixing microorganisms convert atmospheric nitrogen into nitrites and nitrates. These microorganisms are also called as diazotrophs or diazotrophs. They are of two types: symbiotic nitrogen fixing microorganisms, for example, rhizobium and anabina. Then another non-symbiotic nitrogen fixing microorganisms. These examples are Azotobacter, Nostoc and Clostridium. Actually, these symbiotic nitrogen fixing bacteria are having number of examples are there. These see the leguminous plants bearing the root nodules. In the root nodules, the bacteria present, they are useful as the nitrogen fixing bacteria. Then second type, phosphate solubilizing biofertilizers. These are the bacterial species solubilize insoluble inorganic phosphate compounds. For example, Pseudomonas striata and Bacillus uh, polymixa. Besides this, Agrobacterium, then Micrococcus, then Aspergillus species are also uh, phosphate solubilizing biofertilizers. Then third type is compost making biofertilizers. Thus, there are number of biofertilizers. That compost making biofertilizers, composting, that means composting is a natural process and that turns organic material into dark, rich substance called as compost or humus. जे काही डिके झाल्यानंतर उरतं मटेरियल ते द कंपोस्टिंग प्रोसेस इज डिपेंडंट ऑन मायक्रो ऑर्गॅनिझम्स टू ब्रेक डाऊन ऑर्गॅनिक मॅटर इन टू कंपोस्ट म्हणजे खतामध्ये रूपांतर करण्यासाठी देर आर मेनी टाईप्स ऑफ मायक्रो ऑर्गॅनिझम्स फाउंड इन ॲक्टिव्ह कंपोस्ट सच ॲज बॅक्टेरिया फंजाय ॲक्टिनोबॅक्टेरिया प्रोटोझोआ अँड रोटिफर्स. Then next type, that is the Cyanobacteria as biofertilizers. Again, many Cyanobacteria are used as biofertilizers. For example, Anabina, Nostoc and Oscillatoria. There are examples. See, there is the Anabina and Azula. There is the two examples are there. Anabina associated with the plants like Azula and Cycus. There are nitrogen fixing bacteria. Then fungal biofertilizers. These mycorrhiza, fungal biofertilizers, example mycorrhiza, it is used as a biofertilizer. Besides this, first we study one point remaining. There are benefits of the biofertilizers, that is low cost and used as the marginal farmers, used by the marginal farmers, free from pollution hazards, increase soil fertility. Then blue-green algae used as a biofertilizer secretes growth-promoting substances, organic acids, proteins, and vitamins. Besides this, azotobacter supply nitrogen and antibiotics in the soil. Then biofertilizers increases physicochemical properties of soil like texture, structure, pH, water holding capacity of soil by providing nutrients and organic materials. In this way, there is the merits or advantages of biofertilizers. Then fungal biofertilizer. These mycorrhiza used as biofertilizers. There are two types, ectomycorrhiza and endomycorrhiza. It accelerates the ectomycorrhiza. It accelerates the uptake of water and nutrients like nitrogen, oxygen, calcium, and potassium. Mycelium is present outside the root. And another example is endomycorrhiza. Mycorrhizae or rhiza, they are present in cortical cells of root. They form arbuscles and vesicles, so they are called as the vesiculo-arbuscular mycorrhizae, that is commonly known as the VAM. There are also benefits of mycorrhiza, that selective absorption of phosphate, zinc, copper, calcium, nitrogen, manganese, then boron and iron. It enhances the water uptake. It induces the growth by secreting hormones. And it offers protection to the host plant from other microbes by secreting antibiotics. त्याच्यानंतर next, biofertilizer microorganisms. Again, there are number of microorganisms that are used in the biofertilizers, such as rhizobium. Rhizobium is a rod-shaped motile aerobic gram-negative nitrogen-fixing bacteria and containing nitrogen-fixing genes. That is important. It is known as the NIF or NIF, nitrogen-fixing NIF genes. For example, Rhizobium leguminosarum in pea plant. Example with the microorganism, and another one in the bean plant. Pea and bean, these are the two varieties, Rhizobium fascioli. These are the two examples, Rhizobium fascioli आणि किंवा बीन आणि पीमध्ये असणारे स्पेसिफिक बॅक्टेरियाज आहेत. त्याच्यानंतर सेकंड वन इज द अझोटोबॅक्टर. It is associated with roots of grasses and certain plants, and it is free living aerobic नॉन नोड्यूल, मुळांवरती गाठी असणं, मगाशी आपण एक्झाम्पल बघितलं, इथं नॉन नोड्यूल फॉर्मिंग बॅक्टेरिया आहेत, म्हणजे मुळांवरती गाठी त्याच्यामुळं होत नाहीत. तर असे जे अझोटोबॅक्टर आहेत, त्याचं एक्झाम्पल स्पेशली इट इज यूज्ड ॲज ए बायोफर्टिलायझर फॉर ऑल non-leguminous plants, especially rice, cotton, and vegetables. Then third example, Azospirillum. It is free living aerobic nitrogen fixing bacteria associated with roots of wheat and ज्वार. ज्वार आणि व्हीटमध्ये हे जास्त आढळतात. It fixes the considerable quantity of nitrogen in non-leguminous plants such as cereals, millets, cotton, oilseeds, etc. म्हणजे ज्वार आणि व्हीट हे तर मेन आहेतच, पण त्याच्याशिवाय एक्झाम्पल्स आहेत. नेक्स्ट एक्झाम्पल दॅट इज द ॲनाबिना. मग अशी आपण बघितलेलं आहे. इट इज मल्टीसेल्युलर फिलॅमेंटस सायनोबॅक्टेरिया and also forms symbiotic relationship between with certain plants such as coralloid roots of सायकस प्लांट आणि अँथोसिरॉसच्या थॅलसमध्ये आपण ॲनाबिना स्टडी करतोय. इट हॅव स्पेशल सेल कॉल्ड हेट्रोसिस्ट. त्याच्यामधलं एक स्पेशल सेल आहे दॅट इज नोन ॲज द हेट्रोसिस्ट, विच आर द साइट्स फॉर नायट्रोजन फिक्सेशन. Then next example, that is Azolla. Azolla is a free-floating fern, aquatic fern consisting of floating rhizome, that is the stem, and with small overlapping bilobed leaves or roots. Anabenna filaments are present in Azolla leaf, and it is used as biofertilizer in the rice field. राईस फील्डमध्ये जास्त करून ह्याचा यूज केला जातो. अशा पद्धतीनं मायक्रोब्स बायोफर्टिलायझर म्हणून आपल्याला नंबर ऑफ एक्झाम्पल्स स्टडी करता येतात त्याच्यातमध्ये. थँक्यू.